We're very fortunate tonight to have Dr. Ken Huttner. He's a Senior Director of Clinical Research for Ectodermal Pharmaceuticals. He's board certified in pediatrics and neonatology, and he has extensive research experience. He joined Ectodermal in January of 2010. Is that right, Ken?
Exactly.
Okay, we also have Ramsey Johnson. He is the Director of Clinical Operations. He brings 13 years of experience in clinical research and business to Ectodermal, and he joined in March of 2010. So I'm going to go ahead and let Dr. Huttner begin, but I want you to know we'll probably end promptly at 8, a few minutes after, since I was a few minutes late in getting started. And when this is very informal, thank goodness, since I kind of messed up the beginning of the call. We're going to ask, Dr. Huttner is going to give a brief presentation, then we're going to open it up for questions. If you can type your questions in the chat section, I think since we have a lot of people on the call, that might be most convenient, but if that doesn't work for you, you can certainly go ahead and ask your questions. And again, this call is being recorded so that other people can benefit from the information. Thank you for your patience, and Dr. Huttner, please, you can go ahead.
Okay, thank you very much, and thank you everyone for joining. As you can tell from the first slide that we have up here, we're fortunate that after Ramsey and I have been working with Ectodermal for the last 2.5 years, we are now in a position where we have the green light from the FDA to start clinical studies in the XLHED, starting with adults. And we've sent the word out by email and contacts, which, again, we're allowed to do now, describing what the study is like in general. And we've received a lot of great questions about that. So what I'd like to do this evening, working with NFED and with all of you who've called in, is to give you a little description of what the study looks like, how we got to this point, and why we're doing it the way we're doing it, and then open it up to all the questions that you might have that we can try to answer for those interested in knowing more about it and maybe sharing information with friends or family who might be interested in participating. So I
do have one more comment, Dr. Huttner. If you have called in and you have a busy family or a dog barking in the background, could you please push *6 to mute your line while Dr. Huttner is speaking and during questions, so you can still hear everything, but it won't disturb the listeners. Thank you. Go ahead, Dr. Huttner.
Okay, sure. So there's a small number of slides that we'll be presenting are taken directly from the clinicaltrials.gov website. So for those who haven't gone on that site before, it's www.clinicaltrials.gov, and then you can search under Ectodermal or whatever you want to look at, and you can see all the clinical trials involving patients or subjects in the United States. So this will be, again, these are copied directly from that site, so you can see them also. So I'm trying to go to, okay, so this slide is the first, as the top of what you'll see. So what it tells everyone who looks at it is that the study is open, and this is what is called a phase 1 study. It's a safety study, and it's going to be in adults only. This is both males and females with XLHED. And it's open label, meaning that everyone is going to know what they're getting, and they'll all be receiving the study drug, which is EDI-200. It is a multi-center study. We will have 2 sites, and Ramsey will tell you a little bit more about those sites after I get through these slides. And the goals of this study are to provide information on safety and how EDI-200 is metabolized or distributed in humans, which has never been done before. So the next slide, let's figure out how to move this forward. Here we go. Okay. The next slide shows you further study details and talks a little bit about how we're going to do the study. So whenever you design a study for the FDA, they like you to have a primary outcome measures. And for us, the primary measure really is the safety. And by safety, what we do is we monitor everything that happens involving participants, whether it has anything to do with the study drug, which in this case is EDI-200, or not. Somebody falls, somebody, gets a laceration on their finger. Anything that happens, we monitor, and then the safety monitors and the physicians would assess, what might have caused it. So safety is what is the primary outcome, and then we do a number of secondary outcomes, which are just exploratory, just so that we understand a little bit more about aspects of XLHED, which may change, possibly, but we think unlikely, with administration of EDI-200. And you can see on this slide that there are a number of things that are being tested, everything from hair growth to how the lungs work to sweating. And all this is being done both before the subjects start on the study drug and after, with the total duration of the study, which is about 6 weeks. And I see a comment, is there any way to increase the size of this? And I don't know. These are copied directly from that website, the clinicaltrials.gov. So if you're having trouble reading it, and I agree with my eyesight, I am too, I think we'll just have to, you could go to that website and you'll see the exact same information. But I'll talk you through it. So the way that this study is going to be done is to have an assessment done, including a number of laboratory tests, and then individuals will start receiving study drug. And there are going to be 2 groups of individuals in this study. The first group starts at a dose of 3 milligrams per kilo, so they get an IV dose, and they get 5 doses. They're given about every 3 to 4 days, so that'll take about 2 weeks. And after we've done 3 individuals who've been dosed at the first dose, the 3 milligrams per kilo for each of their doses, then there will be 3 more individuals who are called cohort number 2, and they will be dosed at 10 milligrams per kilo per dose. So both of these doses were chosen because they're doses that have worked in the dog model of XLHED. 10 milligram per kilo per dose worked much better than 3, but since we don't know what's going to happen in humans yet, we're covering the range that was used in dogs. So the total number of doses, which is going to be given for each subject, is 5, so again over approximately 2 weeks, and then they'll be followed after that for 1 month. So the total duration is about 6 weeks, and part of that time the subjects will need to be at the study site, and for most of the time they won't, but they'll come back for an infrequent visit. Again, I apologize for the small type, but this is the eligibility. There really aren't too many criteria for being in the study. The goal is for basically young adults, 18 to 40, and adult for the FDA is 18. And because of the amount of drug that we're going to have to use, we had to limit the weights, and it's based on something called the body mass index, but basically it's about 100 to 200 pounds, so not too thin and not too overweight. Now we expect that it'll be important to have information from both males and females with XLHED. The males primarily because the first study in infants with XLHED will be in boys. And having females in the adult study means that there may be studies in infant girls, but also we see that someday in the future, depending upon results of first studies, there may be the opportunity for antenatal dosing, and for this we would need some exposure and safety information in non-pregnant females. So that's why we're enrolling both males and females. There's no fixed number, except in each of the two cohorts we want at least one male and at least one female participating. Now, those familiar with XLHED in women know that as carriers, their phenotype, their presentation can be very variable. And this is related to several different factors, but including in that is the X inactivation pattern, where there will be areas on their skin, for example, where they sweat normally. And adjacent to that, but hard to detect from the surface, there will be areas where they sweat very little. And that's going to be true for any assessment that involves localized areas. So for that reason, when you look at all of the assessments, the secondary endpoints that are going to be done, they're different for men and for women who are participating. So, for example, the men, if they have remaining hair follicles, they'll have a hair study done, but the women won't. And when it comes to assessing changes in sweating, we wouldn't know where to look because of the patches of normal and the patches of XLHED skin, so that won't be a significant part of the study in women either. Some of the studies that are more assessing an entire organ like the lung or like tears or saliva, those will be done on both men and women. So it's a little bit of a simpler process and a little bit shorter for the women who participate. Generally, we want people in good health who have not been on other study drugs for other studies in the previous 3 months. And there's also criteria that are required related to contraception. And again, these are all spelled out clearly, more clearly when you go to just the website for clinicaltrials.gov. I see a question popping up about adult study for individuals without HED, and I'm going to hold that for just a second until I get through the slides, and then we'll talk about why we're doing this the way we are and what we're looking forward to in the future. There are exclusion criteria, particularly women who are pregnant or individuals who have active hepatitis or HIV or might be sensitive to some of the procedures that are going on, and you can see them listed on the slide deck. Okay. The next slide, the print's a little bigger, but just gives you the location of the two sites. Ramsey will talk a little bit more about these. One is in California at the University of California, San Francisco, and one is in Cincinnati. And all the contact information you would need for any questions, you can find both phone numbers and email addresses, again, on the clinicaltrials.gov website. I think that's it for the slides. And let me just give, before we open the floor to all questions, let me give you a little bit of a background of the discussions that we've been having over the last couple of years with the FDA and how we got where we are. So I won't make this too long, but just it might address some of your questions. So when we started this, working on this program, there had already been a meeting with the FDA to talk about how the study should be done. And in that first meeting, there was a discussion of treating newborns, and everybody seemed to realize that the target population was going to be newborns, if not earlier, and nobody really got on this subject of should we do something before treating newborns directly. And I think that is one of the key issues that Ramsey and I have been discussing with regulatory agencies like the FDA and BfArM in Germany, as well as key opinion leaders and the sites where we might do the study about what should we do to feel as comfortable as we can for a drug like EDI-200, which we want to target to newborns, as vulnerable a population in theory as one could find. And as the discussions have progressed, then it's really come back to would we feel more comfortable if there was some adult data? And in discussions with the FDA, it was clear that they would feel more comfortable, that there are no other drugs that they have under development that are going directly into newborns, not just pediatrics, but going directly into newborns. The German regulatory authorities felt exactly the same way. And so it became pretty clear that adult data, although, again, adults are so different from neonates, and that every drug we use, they have different dosing and different concerns, but it seemed that experts in the field and the sites and the regulatory agencies all would feel more comfortable with adult data. Now, it didn't have to be a large adult study, and we had some discussion about who would be the most appropriate adults to do the testing on. And we went back and forth on this. In the end, we felt that we wanted to get the most accurate information that would reflect on the genetics of the newborns. So if the newborns were going to be HED, that they were going to have EDA deficiency, that we would learn the most by involving adults who also had XLHED and had EDA deficiency. So at the end of the day, that was the decision from Ectodermal. We had a discussion with the FDA, and they were fine with that. So that's how we came to the idea that it would be adults, that many have requested, and that it would be adults with XLHED that was documented by genotyping. So once we had that in mind, and we all come to an agreement on that, Ramsey and I have been working on the study design, and our key message is that we wanted to have a study design where what we were doing in adults was as similar as possible to what we would do in neonates. We really wanted to set the stage. And so we outlined how this might go in neonates, and we based that on all the information we had from Maggie Cassell and her dogs, because they're the closest model to what we would do in the clinic. And based on that, we knew we wanted to go 5 doses. We knew the doses that we were shooting for were the ones that we knew worked in dogs. That's no guarantee that it's going to work in humans, but that's the best place we could start, and have some background support for what we were doing. So that's why we would start with the 5 doses, the 3 milligrams per kilo per dose, or 10 milligrams per kilo per dose, and over the same time frame that it was given safely in the dogs. Now, because of the concentration of EDI 200, which is a protein, it's a biologic, and it's it's 2 pieces of human DNA that are fused to each other, and so it's all human material, but it's 2 pieces that aren't normally found linked together. So it is a novel molecule, but it's all human. And so because of its complexity, it has to be given intravenously. It can't be given as a sub-Q injection. It can't be given orally. And so for that, intravenously, it's got to be given slowly, and that's built into it. And when we talked to the FDA about doing it, we said, since it's the first humans that will receive it, we will do all the appropriate safety studies that the FDA requires before we submit to get the green light. But also, every time we give a dose, we will monitor that individual at the site for 24 hours after the dose. So that's the type of drug, that's how we're going to give the drug, and that's how we would give it to neonates as well. We've set it up so that we're doing 1 or 2 individuals at a time. We're going to get safety data from that 1 or 2 individuals before the next group comes in. So it's going in a sequential manner, and that, again, provides a level of comfort and safety to all those who will be monitoring, and that includes the study site, as well as myself and Ramsey coming at Ectodermal. And in the end, we will have an independent safety monitoring committee, which receives all the information live, so they get real-time reports on laboratory studies and anything that goes on with any patient that could be of any concern. So that's the way the study will go on. And I see some questions about the entry criteria. So part of what takes a long time to get EDI 200 ready to work is that it's hard to grow the cells that make EDI 200 and to isolate enough material for the adults. So neonates is not a problem for a 3 and a 4 kilo baby, but at these doses it takes a large amount. So just for example, the vials that we have will have this EDI 200 frozen in have about 2 mils in them. So for each dose of the adults, we're going to be opening over 100 vials and having to pool it together. So for the pooling, number of vials, and the amount of material, we had to put a limit on the upper weight. So, that's for better or for worse. That's why we have the upper limit. And if you think you're near that upper limit or near the lower limit, to make sure that you're not too underweight, we have a formula that we can use to calculate that. And again, if you email Ramsey with any question about the entry criteria, he can give you that information and let you know, whether you qualify for it or whether it would be an issue that would disallow you from participating. Now, I'll get off shortly. The first study is scheduled for 6 adults. Again, a small number, but a number that at least, for the regulatory people, they felt, okay, we're going to learn something from the 6 adults that's going to make us feel more comfortable with neonates. Now, 6 may not seem like a large number to recruit, but we do recognize that this study is scheduled to run starting later this year. It's a tough, difficult time of year. We're asking individuals to come in for several weeks away from their family. So we are trying to make sure that we have enough that we have individuals, multiple individuals who might be considered when the actual time comes. We know that people's lives change at the last minute, and so we want to make sure that this actually goes off. So that's sort of the sum of how we came to this point. And I'd like to pass it on to Ramsey for a minute to talk briefly about where we stand with the sites, which order the sites are going to be used at, can you go to either site, how many individuals do we want signed up, and then we'll be happy to answer any questions.
Okay. Thanks, Ken. And thanks to everyone on the phone and to everyone that's emailed me. We've had a great response since Mary sent out the notification, and it's been a big comfort to Ectodermal to know that there are so many people out there that are willing to participate and that are interested in finding out more. So as Ken mentioned, we will have 2 sites. One of the sites is in a clinical research unit that is right on the campus of the University of California, San Francisco Medical Center. So it's located right there in the hospital, and the principal investigator at that site is someone that we have worked with before, and that some of you that are on the call or that are interested in participating may have even met at one of the natural history studies that we did at UCSF last year. So that is one site. The other site is in Cincinnati, and it is what we would traditionally call a sort of standard phase 1 unit. So it's not in a hospital, but it's in a unit that does clinical research and these types of studies all the time. This is what they specialize in. They are attached to a hospital in Cincinnati, which is good, for access to varying specialties, and should anything happen and someone needs medical attention, they're right there. And the principal investigator there at that site is someone that has done lots and lots of research, and so is very experienced. We are going to try and limit each cohort to one specific site. So Ken mentioned there'll be 2 cohorts, one at a lower dose and one at a higher dose. The way it's looking right now is we would probably like to do the first cohort, cohort 1, at the site in San Francisco, and then that will be followed by cohort 2 at the site in Cincinnati. Of course, all that is subject to change still. It really is going to depend on who we have that steps forward that expresses interest and which site makes sense for them. And as I mentioned, we've gotten a great response since Mary sent out the email. I do have some participants that are quote-unquote confirmed in that they've confirmed with me via email that they want to participate, but we still have quite a bit of time. Things change, schedules change, and so those people in the end may not be able to participate, but they have confirmed to date. That means, though, that we still have a couple of spots open for people to confirm now that they can and want to participate. But even more important than that, I think, is to have a pretty large pool of backups should someone that's confirmed end up not being able to participate. And so that's why it's great to have this call. It's great to continue to get the emails with people that are interested in participating. And I would encourage people to email me and to continue to email me if they're interested, even if all the spots fill up. Again, it's very important that we have backups so that we're not delayed if one of those confirmed participants chooses not to participate for whatever reason. And then just a little bit on sort of how long you'll be expected to be away from home. I know that's a big question that I've been getting over the course of the last couple of weeks. And so I would say that unless you live in the immediate vicinity of either Cincinnati or San Francisco, then you should expect to be away from home for about 2.5 to 3 weeks. And so by the time you'll need to come a couple of days in advance of your first dose for some baseline evaluations. Then you would get your first dose, and you would need to stay overnight at the clinical research unit. Then Ectodermal and the research unit would help coordinate hotel accommodations sort of near the clinical research site for you. Then you would come back into the unit for your subsequent doses 2, 3, 4, and 5. You would stay in the unit for 24 hours after each of those doses, but then you could go back to the hotel, and you'd stay at the hotel in between those doses. Once the doses are done, there's a couple of follow-up visits that would happen in the days immediately following the last dose, and then you'd be free to go home. And then you would need to come back for a couple of follow-up visits after that, but those could be day trips or at the very most overnight visits where you'd stay at a hotel and have some evaluations performed. So I know that 3 weeks sounds like a long time, but there are a few periods in that 3 weeks where you'll have a couple of days in a row where you won't be required to come back to the research unit. And so we could certainly work with you if you needed to go home for that time period and then come back. We're open and we're flexible to working around whatever works for your schedule. Okay. I think that's it for me, Ken.
Okay, just to mention that, of course, all the expenses involved in transportation to the site, transportation back home, and the cost while you're in either Cincinnati or at the site in San Francisco are covered by Ectodermal as a sponsor.
Yep, that's right. And there is some compensation to the subjects for participating above and beyond having your airfare, your hotels, and your meals covered while you're in the vicinity of either one of those research units as well. So you should keep that in mind too. because it will require you most likely to take some time off from work, and Edimer is aware that it's an inconvenience, and so you'll be compensated during your participation.
Okay. So, at this point, why don't I try, if it's okay with everyone, why don't I try to address some of the questions that are coming on the chat box here.
Great.
And then I can follow that up. So, one question, did we do an adult study for individuals without HED? And as I explained before, we had a choice of which one to do, and we thought the most appropriate one was the one in individuals with HED. We don't have any plans at the present time to do any other adult studies, but of course, if something surprising and positive comes from the adult study with all the assessments that we're doing, then we would take another look at that. A question, do you expect that the drug would be needed as a lifetime treatment? So we know that in general, development of sweat glands and hair follicles and teeth takes place or is initiated over a relatively short period of time. And that's borne out by both the results in mice and dogs with XLHED, where a short course, sometimes a single dose or 2 weeks of dosing, is all it takes. And once the pathway is activated, then everything proceeds to mature, and the effects are things that are seen all the way through adulthood in both animals, in both the mice and the dogs. So, we recognize, and I get asked all the time, does that mean that, humans that take much longer to develop will also have a very long sustained response to one dose or a short course, and we won't know that until we get into the clinic. But it's very promising in the animal models that, at the right time, with the right dose, a short course can give a lifelong benefit. What we get asked the most, and it's a very appropriate question, is, what are the predicted side effects or adverse effects? And, the top line answer to that is that since this has never been given to a human of any age, then, we don't have an answer for any side effects that are predicted. We just don't know. But as part of the effort to get a new therapy like EDI-200 into clinical studies, there's a well-defined path that the FDA has, and they have in Europe also, that is used to produce as much safety information as one can from animals. So we have followed that path, and I can tell you in general the way that the FDA likes to do it is for any new medication, any new therapy, they like you to use doses that are at least as high, if not higher, than you would expect to use in humans, which is what we did. And they would like to do it in at least 2 species, one of them being a rodent, like a mouse or a rat, and the other not. The problem that drug developers have had for decades is the following, that the molecule that we're developing can be found in mice, it can be found in dogs, it can be found in humans. So the sequences that we've chosen to use are ones from humans. So what that means is it's probably safest that we can make it for humans, but it's a very foreign molecule to other animals. And when you give a foreign molecule like EDI-200, the human form, in large doses, and you give it very quickly IV to animals like adult mice or adult dogs, what you see very consistently is that they develop what looks like an allergic-like reaction. It's not an allergy, but but they get a rapid response like an allergy, and the more doses they get, then the more trouble they have. So that's the problem that the FDA and many drug developers have worked on, is what to do when you're trying to make something like EDI-200 as safe as you possibly can for humans. It means it's less well tolerated by adult animals. And so we found that to be true, that when we give high doses, the doses that we're using of EDI-200 by an IV injection into adult mice and adult dogs, they develop exactly what other people report. They have these allergic-like reactions, and that's very clearly associated with the rapid injection of a foreign molecule. So when this happened, and that was about a year ago, we sat down with experts both that work with the FDA and people who used to work at the FDA, and it's very straightforward. They said that, this is not enough safety data to allow this to go forward, in their opinion, that we would need to show that if we deliver EDI-200 to a species closer to humans, and we deliver it the exact way that we're going to deliver it in the clinic, which is by slow IV infusion, that should be sufficient to know, to reassure the FDA one way or the other that we have the safety data to proceed into human trials. So based on that, we took 6 months and a fair amount of time, effort, and money to do that exact study. It's a safety study in a third species now, and that was in monkeys. So we have a primate, and the sequence is very similar between this molecule, the sequences that we use in EDI-200 and the sequences that are in monkeys. And these monkeys got a small IV like we're going to use in the adult humans, and we gave the infusion very slowly, just like we're going to do it in the clinic. And I'm happy to report when we did that, there were no adverse effects. There was nothing seen clinically in the monkeys, no change in their behavior. They had laboratory tests. They had electrocardiograms. They had samples looking at every part of them, and they were followed in some cases for 6 doses, so that was 3 weeks, and then some weeks afterwards, and everything went beautifully. So that was great, but again, we put all that information together from the reactions in the mice and dogs and from the safety in the monkeys, and we packaged all that in a 10,000-page report. I think it was something along those lines, and we sent it to the FDA when we requested that we would like to start the adult studies. And I'm happy to report in the spring that they reviewed all the data and gave us the green light that, to the best of their ability and our ability to judge from animal studies, that there were no particular adverse effects that would be predicted, and that as long as we went about doing the study the way we're doing it, we start with one individual, we start with a lower dose, we have a safety monitoring committee. As long as we do all those things, then they believed it was safe to proceed. So that's what we've done, and we've done the best that one can do in an animal. An animal result is no guarantee of what will happen in humans. Again, this EDI-200 administration will be done slowly in a setting where there's medical care right there, so that if anything, if there's anything that goes on, we know that sometimes people have allergies that they don't know about, and there's no way to predict. But there's no reason to suspect, based on everything that we've known so far, that there would be any predicted adverse effects. So I just want to stop 1 second there and just see if there's any clarification that anybody would like on that information.
There is about 15 minutes left of this call.
Okay. All righty. Then I'm going to just I'll move through some other questions, and you just stop me if you have anything else that you want to share, want me to clarify it more. Any intentions to make it available to adults once it's approved? That really depends on what we find when we do this adult study. We're not going in with the expectation that that there will be any benefit, but we are going to be measuring everything we know how to measure, and if we are surprised on the upside, then we would reevaluate whether there would be an indication that it might benefit adults. We learn more and more from individuals like those who are listening on the call that there are so many issues in adults that are lifelong, and so we can't rule it out. But right now, we have no evidence that we'll be able to change anything about XLHED once you're through, your adolescence. But again, that's something that we will find, and we will share all that information with the participants and with the FDA and, of course, the NFED once the study is completed. We know where the studies take place. Now, again, I want to emphasize that it's been my goal from the beginning to mirror the infant study and the adult study, mirror them in the same number of doses, the spacing between the doses in terms of number of days, and the actual dose for each one. So it's based on their weight, milligrams per kilo of body weight. Now that makes sense as long as the metabolism turns out to be similar in infants who get EDI 200 and adults. So we have data from some newborn dogs and newborn mice and some adults to say that we expect there will be similar metabolism. But once we start, we'll gather the data first from the adult humans who will participate in the study. So they have safety data, but they also have blood tests that tell us what the peak level of EDI 200 is after they get a dose and how long it stays in their body. And from that, we'll be able to see whether it seems to be consistent in humans as it is in monkeys and dogs and mice, what the metabolism is. If there's any suggestion that the metabolism might be different, then we might use that, and we should use that information to adjust the dose in the neonate study. But our intention and the way we've designed it at this point is that it's going to be a mirror, that we're going to take the same doses, same number of doses, same timing in adults, and translate it directly into what we would do in the newborns. We have no expectation of potential long-term side effects for humans. We have had some questions about whether adults would all of a sudden grow a new set of teeth. We see that. There's going to be new biology written, but at this point, our experts in dentition feel that we're destined not to have more than the primary and the adult teeth, and that we would not initiate the growth of any tooth buds. So that would be quite an unusual surprise. There's nothing we could do in an animal model to tell us that. So there's really nothing we can do at this point. The data we have in animals for safety that the FDA requires is all done in animals that do not have XLHED. So these are wild-type animals, and that's just the way that they ask that the information be generated. Now, even though there are no specific adverse effects that we are anticipating, one can have an allergy to any protein. So it's something that's always possible. We don't expect that it would be any more possible with this molecule than dozens and dozens of protein therapeutics that are used in practice all the time, but it's something that's monitored both in terms of blood pressure and other vital sign changes, but also as part of this there will be testing of immunogenicity, which means if there's evidence that antibodies are being generated, like a vaccine effect almost, and we'll be testing that in the blood of the adult subjects through the blood samples that we draw during the course of the 6 weeks that they're involved. And again, one of the beauties of this approach is that it's a very short term. It's not like multiple boosting therapies every month. It's a short term. We're looking at the 5 doses over 2 weeks, and then you're done. I see a question about the teeth in dogs. It's been published how improved the teeth are in dogs. They are as close to normal as we can, in terms of function, as we can see. Although I wouldn't say that they are, absolutely identical between the treated and the wild type. And if you go onto our website, we can get you access to the papers where they show pictures. But again, they're very close to a fully functional, and certainly they have a restored normal number of teeth. A question about pre-testing. There's no way to predict the allergic potential to a new protein like EDI-200. So there's no one test that will tell that. There are individuals who can be very allergic, and they may or may not be more sensitive to it. So, that's just going to have to be a concern with individuals who tend to be more prone to allergic reactions, but there's no way that that's 100% predictive that they will or will not react to this molecule. Again, a question coming in about what the short-term or long-term impact on ectodermal dysplasia would be in the adults. All of our evidence suggests that there was no predicted benefit, but we see no prediction of any specific adverse effects either. This is really a safety study that's demonstrating safety for the future and for the future generations of XLHED patients who we are targeting EDI-200 for. This molecule shouldn't have any impact on DNA, so there's no reason to, it's not going to remove any risk that your kids would have XLHED, and it shouldn't have any negative impact on future offspring either. Just a follow-up question here as I see the safety studies that were done in adults were done on wild-type mice that are adults, wild-type dogs, and wild-type monkeys. So none of those that were done in the safety portion for the FDA had XLHED. But Pascal Schneider, who was the first investigator along with Olivier Gay to do the mouse studies with EDI-200, and Maggie Casal, who's done the dog studies, they have dosed a large number of animals, but those animals are newborns. So they don't have adult safety data. The adult safety data has come from the work we did preparing for the FDA. In general, studies like this phase 1 safety study are short-term. There's, if there are any safety or health concerns up to 30 days after the end of the study, those are included in the record, but there's no provision for long-term testing in this current study design. Again, going back to the dosage that's being used, the dose that we're doing, again, is not the set amount like milligrams or pounds or ounces of material. It's all based on weight. And the dose that was used in the animal studies, particularly in the dog, was between 3 and 10 milligrams per kilo. The dose that we'll use in the adult human study, this first study, will be between 3 and 10 milligrams per kilo. And if the results suggest that it's behaving normally and the metabolism is consistent across all the species, we will use exactly the same doses, 3 to 10 milligrams per kilo, in the newborns. The protein, as we've been able to find in the animals, in general lasts less than a week in your system. So we would expect, and again, you can be surprised in individuals, but in general, that it would be totally out of the body within 7 days. I see some questions about newborns, and I'm going to pass on how many we're going to be needed. We're working on the study design, and when we have that finalized and submitted to the FDA for their review, then we'll be happy to share that. The FDA does recognize for rare disorders that there are not an infinite number of patients, and for a study that's going to enroll newborns, that it'll be a relatively small number that we'll start out with. Okay. Last question I think I'll have time to get to is what's the time frame between when we will have the adult data and when we would start the newborn? So current plans, and again, this certainly depends on having individuals who volunteer to work with us in the safety study, would be to have the adult study completed and all the data in late spring of next year. Now, if all of that looks as reassuring from the safety point of view and the metabolism point of view as we think it will, and we hope it will, and we expect it will based on all the other data that we have from our safety studies, then at that point, certainly we believe that that would be a time for the FDA and the European agencies to consider giving us the go-ahead to start treating newborns. Whether we can do that earlier is certainly unclear. We will be in discussions with them, and we've been offered the chance to have those discussions through a process called, fast tracking, where they're letting us have as many meetings as we can to move this forward quickly. And so we'll discuss with them in this fall and winter as the adult study starts to see how quickly they're going to feel comfortable and making sure the paperwork is into them so that when the data is there and everyone has a chance to see the safety, which we expect from the adults, and the metabolism rate, then we would have the right information to inform us how to do the study in newborns as safely as possible. So, next year would be the year, but until we actually see the data, we can't share it with the FDA, and they can't review it. So the first half of next year will be a very exciting time for us. So I think we're running up to about, I think it's 8 o'clock Central Time.
Well, I want to thank everybody for joining tonight's call. I really thank Dr. Huttner and Ramsey for agreeing to do the call or being so anxious to do the call on such short notice. We had so many questions, and we thought this would be the best venue for you to have all your questions answered. They're always happy to answer your questions. You have Ramsey's email. You also have mine. I also want to apologize for the technical difficulties we had at the beginning of the call. I'm not quite sure why I got bumped off as the chairperson. And I appreciate Kelly rescuing me, going in the back door and booting me off and signing in as the conference chair. You will get a link to the recording in the next couple weeks, and it will be available on our website. I greatly appreciate your time and attention, and thank you all.
And have a good evening, everyone.
Have a good evening. Thank you.
Thanks,
Good night. Thank you. Good night.